Hi, this is our Central Dogma modeling project.
DNA is a nucleic acid that is built up of the monomers called nucleotides. In DNA nucleotides, there's a 5-carbon sugar called deoxyribose. On the 5' carbon, there's a phosphate group attached, and this forms the backbone of the DNA. The 3' carbon is then attached to the next nucleotide, forming a phosphodiester bond. On the 1' carbon of the DNA, the nitrogenous base is attached. DNA has 4 bases: adenine, thymine, cytosine, and guanine. These are categorized into pyrimidines and purines. G and A being the purines and T and C being the pyrimidines. Due to the base pairing rules, pyrimidines bind with purines, so adenine always binds with thymine, and cytosine always binds with guanine. These bases are bound through hydrogen bonds. A and T have 2 bonds, and C and G have 3. DNA replication is a semi-conservative process, as each new strand of DNA has one parent strand and a newly synthesized strand. Due to the base pairing rules, the parent strand can act as a template, so that a new complementary strand can be synthesized. To start the process, the parent DNA is unzipped, or the 2 strands are separated by the enzyme helicase. This enzyme breaks the hydrogen bonds between the nitrogenous bases of the 2 strands. A protein then binds the strands to make sure they do not rebind. As the DNA is unwound, a replication fork forms, where the DNA is opened. Topoisomerase is an enzyme that is behind the replication fork that helps relieve the stress of unwinding the DNA. DNA polymerase is the enzyme that actually carries out the DNA replication, adding 1 nucleotide at a time. DNA polymerase can only add onto an existing chain. It cannot start a new strand of DNA on its own. Therefore, a small piece of RNA called a primer is attached to the parent strand, so the DNA polymerase can build off of it. The parent strand with its 5-carbon end facing towards the replication fork is called the leading strand. Since DNA polymerase can only add nucleotides in the 3 direction, DNA polymerase can continually synthesize DNA on the strand. On the other hand, the antiparallel strand across from the leading strand is synthesized discontinuously, so it's called the lagging strand. This is because the nucleotides can only be added in the 3 direction, which is facing away from the fork. Therefore, the helicase enzyme unzips a little of the DNA, a primer is added, and the DNA polymerase builds the chain away from the fork. Fragments in the lagging strand are called the Okazaki fragments. This process begins again to create another Ozaki fragment. Because each time this process occurs, a new primer is added, there are many chunks of RNA in the lagging strand. As DNA polymerase moves on to the next fragment, it removes the RNA primer from the last fragment, as there cannot be RNA chunks in DNA. Then an enzyme called the DNA ligase joins the 2 adjacent Okazaki fragments to form a continuous strand of DNA. From there, the protein that was keeping the strands separated detaches, and the DNA rewinds, resulting in 2 strands of DNA. Lastly, while each nucleotide is added, DNA polymerase has a property that allows it to proofread its work. When a wrong base pair is added during DNA synthesis, DNA polymerase can recognize this mistake and replace the wrong nucleotide with the right one. This ensures that there are little to no mistakes in the DNA, as mistakes cause genetic mutations. Since DNA is copied and passed down through generations, having genetic mutation can be lethal to the cell and even organism.
In DNA replication, DNA can be mutated through base substitution, base deletion, and base insertion. For example, if one base pair in DNA is swapped for another base pair, and it is not picked up by DNA polymerase, this is called base substitution. If this happens on the hemoglobin beta gene, it can cause sickle cell disease. Base deletion takes place when base pairs in a strand of DNA are removed or deleted from the molecule. For example, with the George syndrome, part of the DNA in chromosome 22 is deleted, which leads to immune system issues. Lastly, with base insertion, extra nucleotides are added to the DNA strand. Huntington's disease is an example of this, with over 40 repeats of the bases CAG added to the chromosome 4. RNA is a nucleic acid that is built up of monomers called nucleotides. In RNA nucleotides, there is a 5-carbon sugar called ribose. On the 5' carbon, there is a phosphate group attached, and this forms the backbone of RNA. The 3' carbon is then attached to the next nucleotide, forming a phosphodiester bond. On the 1' carbon of the RNA, the nitrogenous base is attached. RNA has 4 bases: adenine, uracil, cytosine, and guanine. Due to the base pairing rules, pyrimidines bind with purines, so adenine always binds with uracil and cytosine always binds with guanine. RNA is a single-stranded molecule, but when it is transcribed from DNA, it follows these base pairing rules as a hybrid molecule. These bases are bound through hydrogen bonds, A and U having 2 bonds and C and G having 3. In eukaryotes, transcription occurs in the nucleus, whereas in prokaryotes, it occurs in the cytoplasm. Transcription is the process of reading DNA and synthesizing an RNA molecule from the DNA. Transcription is performed by an enzyme called RNA polymerase. RNA polymerase synthesizes an RNA strand by pairing complementary RNA nucleotides to a strand of DNA. There are 3 stages in transcription: initiation, elongation, and termination. During initiation, the RNA polymerase binds to the promoter gene, and then RNA polymerase separates DNA strands. Promoters are regulatory regions in DNA where RNA polymerase and associated proteins bind to the DNA molecule to start the process of transcription. The strand that the DNA binds to is called the template strand. The bases on the template strand are complementary to those on the RNA that is being transcribed, making the RNA a reverse complementary strand to the DNA template. The other DNA strand, called the non-template strand, has the same sequence of nucleotides to the RNA, except T is replaced with U. During the next step, elongation, RNA polymerase reads the template 1 base at a time and builds an RNA molecule out of complementary nucleotides, making a chain that goes from the 5' end to the 3' end. RNA polymerase does not have the proofreading function that DNA polymerase has, so mistakes are more common. Lastly, in termination, there are sequences in the DNA called terminators that signal the RNA polymerase that transcription is complete. The RNA is then released from the DNA, RNA polymerase unbinds, and DNA then rewinds.
Translation is the process of decoding mRNA to build a polypeptide that contains a specific series of amino acids. After being synthesized in transcription, mRNA travels to ribosomes to be translated into proteins. Ribosomes have a large subunit and a small subunit, and are made up of RNA, which is ribosomal RNA, and proteins. In both prokaryotes and eukaryotes, translation takes place in the cytoplasm of the cell. Ribosomes read mRNA in successive groups of 3, which are called codons. Codons code for a single amino acid. For these codons to be translated into amino acids, tRNA is used. tRNA contains around 70 to 90 nucleotides, and hydrogen bonds between these bases causes it to have a clover-like shape. The bottom of the tRNA contains 3 bases, which are called anticodons, as they are complementary to the codons on the mRNA strand. These anticodons determine which amino acid the tRNA carries. Translation can also be divided into 3 different stages: initiation, elongation, and termination. In initiation, protein initiation factors bind to the small subunit of the ribosome or the mRNA itself. The small subunit of the ribosome is used to hold the mRNA in place. This formed complex moves down the RNA strand until it reaches the AUG codon, which is the start codon. Once it reaches the AUG codon, the large subunit of the ribosome joins the complex. The initiation factors are released, and the first tRNA molecule is attached. tRNA molecules attach the mRNA on one end of the molecule and carry the amino acid on the other end. This first tRNA molecule carries the anticodons for AUG, which codes for the amino acid methionine. The next step, elongation, is the stage where amino acid chains become longer and a polypeptide is formed. Ribosomes have 3 spaces for the tRNA to bind, so after the initial tRNA binds, the next codon is read and another tRNA binds the adjacent space. Through a dehydration synthesis reaction, the methionine amino acid is connected to the next amino acid, forming a peptide bond. The ribosome then shifts one codon to the right, releasing the tRNA attached to the start codon. The next tRNA is attached and the peptide chain grows. This process continues for the rest of the mRNA strand, forming a much longer polypeptide. The last step of translation is termination. Termination begins when the ribosome reaches one of the 3 stop codons. Stop codons do not have a corresponding tRNA molecule, but instead a protein release factor binds the ribosome. This protein causes the bind between the tRNA molecule and a polypeptide chain to break, creating the carboxyl end of the protein, and thus completing the chain. Once the protein is released, the small and large subunits of the ribosome disassociate from the mRNA and each other, and therefore translation is completed.